Okay. Oh, click on this. There we go. There's my clicker. Okay, so Monday morning. What a morning. All right. We have been talking about cell division. Remember, the cell cycle is basically about the life of the cycle. On Friday, we talked about, and let me just, back it up. So remember, on Friday, we talked about the cell cycle. G1S and G2 are all part of interphase. That is where this just shows how much of the time the cell spends of its life. It spends most of its time in interphase. We looked at what happens during G1. Remember, it doubles its number of organelles. In S, that is when DNA synthesis or DNA replication occurs. It makes a copy of its DNA. Because remember, each of the two new cells at the end are going to need a full copy of that DNA. Remember, S phase is when DNA is replicated. That could be a star question. So just memorize that. All right, then G2, it gets its structures ready for cell division. Okay? Then, when the cell needs to divide, okay, it will go into mitosis. Mitosis can happen very quickly, within 20 minutes, sometimes more, sometimes less. But mitosis is the division of the nucleus of the cell. And then it goes through cytokinesis. Why did it do that? Okay, then it goes through cytokinesis, is where the rest of the cytoplasm divides. And then you end up with two brand new cells. Okay, so having said that, I'm going to have to readjust my screen. I messed it up. No. Sorry, everybody. Okay. Now let's see what happens. Okay. So it says, let's break down mitosis into the individual stages. And you really need to be aware of what's going on in the individual stages of mitosis. That would be prophase, metaphase, anaphase, telophase. We remember that with PMAT. Okay? PMAT. And then, of course, when we add cytokinesis to that, Riley told me that you can remember, pass me a taco chief. Because it's prophase, metaphase, anaphase, telophase, cytokinesis. Pass me a taco chief. But anyway, PMAT is how we remember just the phases of mitosis. Okay. This takes us through interphase, which we're still looking at what happens in the G2. These little centrioles up here at the top, okay, those are important. Those are going to move to the opposite ends of the cell, and that helps with the cytokinesis, okay? All right. Anyway, during this time, G1 phase is where there's a lot of intense biochemical activity. Once again, it's going through what happens. All right, so here's where I want to start for today's lesson and what's going to be in your assignment for today. All right, remember, interphase includes G1S and G2. It is the time preceding or before mitosis. And here are the 4 stages of mitosis on your screen. That is like one of the first things you have to answer. Prophase, metaphase, anaphase, telophase, PMAT. And then all we're going to talk about for the rest of the day and what will be in your assignment is going to be prophase. What happens during prophase. All right. So, this screen right here is where you're going to get the meat of the information that you need to have for your assignment today. Okay, first of all, the chromosomes coil. Okay, these are the chromosomes. They thicken up and become really distinct. And they become visible. Before an interphase, you will not be able to see the chromosomes. Okay? But when it goes into prophase, the part of mitosis, the division of the nucleus, these chromosomes thicken up and they get to where you can see them. They become visible. Okay? That is one of the things that happens during prophase. The nucleolus, which is a little, it's inside the nucleus. Inside the nucleus is a nucleolus. Well, it disappears. You can no longer see the nucleolus. Okay? It also says the chromosomes double throughout their length, so the chromosomes get longer. Each half of the double chromosome is a chromatid. We went over that, remember, last week. Notice the chromosome up here, they're joined at a centromere, and each one of those is a chromatid. It takes 2 chromatids to make up a whole chromosome. The chromatids are connected by the centromere, once again. The centrioles separate. Those are these little golden little deals up here at the top. So in the last picture, it showed them together. Now they're really they start to separate and go to opposite ends of the cell. And these little spindle fibers, this just calls them spindles. But I want you to know that they're also called spindle fibers. Okay, they start to form. And anyway, that's what's going to help the chromosomes separate, and you'll see that as we go through the other phases. All right. And those little spindle fibers are also made of what we call microtubules. That's what they're made of. And that pretty much covers all I want to go over today. So we have talked about the first phase of mitosis, which is prophase, and we've talked about what happens during prophase. So in some of the STAR questions, you might have to they might ask you about, well, which phase of mitosis is that? And the main thing they're probably going to ask is that the chromosomes become visible, the nucleolus disappears, and maybe the centrioles, or the centrioles up here at top, they appear and start to move to the opposite ends of the cell. And so that's it for prophase, the first part of PMAT. So I'll go ahead and get your assignment activated, and y'all can get busy.
Oh, hell, okay. He'll